And thank you, Mary Lou, for letting me come once again and not telling people that I'm boring, because it looks like everybody came back after lunch. I want to take a few minutes to kind of talk about where the Blepharospasm Society has gone before the BEBRF and after the BEBRF. And then I want to take some time to challenge you to say, where do we do next and where should we go? So I'm going to talk a little bit about the definition of dystonia. I'm going to talk about the history of blepharospasm, diagnosis and imaging, pathophysiology, genetic factors. I'm going to talk about treatment. I'm going to summarize the work of the foundation for the first 30 years that they've been providing grants, and then I'm going to challenge you. And if you look in that bottom right-hand corner, I've tried to go through and list the people's names who received grants in the certain areas of all of these areas. And you've seen similar slides to this all day long, but I thought it was important for you to see how money in the BEBRF has been spent for for research. But if you want to skip my talk, the website is so much better in terms of gathering information and what has happened in the Blepharospasm Society. And so that's what that looks like. So if you type BEBRF into your search engine, that's where you can go. And I urge all of you to look at this. I urge all of you, when you meet people who might have blepharospasm, to share the website with them. This is a place to go for information. I guess one thing I wanted to add that I thought of during our wonderful substitute talk is that Dr. Oberg needs to think about how to write that up so we all understand it. And I believe that we ought to try to find a constellation of people who do partial myectomies well, just like we find people who are dystonia doctors well. So way back in 1984, the DMRF came up with a diagnosis for dystonia. And there it is. They came up with it all by themselves. One of the things that Mary Lou has been very good at doing is sticking to her guns, but saying we all need to play together. Is Diane Trong still here? Yeah. So stand up, please, Diane. So Diane Trong is a dear friend to the BEBRF, dear friend to Mary Lou, and is head of the NSTA, the National Spasmodic Torticollis Association. Thanks so much for coming to be and support us. So many years ago, maybe 8 or 10, these groups began to get together to really refine the definition of dystonia. We don't quite have squeezing back in there or blinking back in there, but I believe we're beginning to move to a more all-encompassing, diverse definition for the multifactored and presentations of dystonia. And I think that it's an important thing to acknowledge and notice that the change in the definition would not have happened until there was a cooperation between all of these dystonia societies. And it's important for people to have a common place to go with common definitions. Vocabulary, just like your teachers told you in grade school, is important. So many things in medical science, and particularly in neurology, is beginning to develop and collect information to begin to figure out how to codify it. And so if we look at where we started, and Dr. Lee showed the chloroform paper this morning, but that's the first report of blepharospasm when you look in PubMed. In 1857, somebody was able to open their eyes a little bit before they passed out from chloroform, and that was a treatment. Then it was described as a point of hysteria, which persists to this day. Then there was a surgery described, a very brutal surgery, much like the early surgeries, a facial nerve resection. In 1956, an essential blepharospasm was described. Myectomy was then perfected a bit, not like Dr. Anderson's surgery, but a bit. Another neurectomy. And then in 1970, an important thing happened. Dr. Rao talked about that a little bit when he talked about looking at patients who were with Parkinson's disease who were given high doses of levodopa and they had eye blinking. That means dopamine can have a factor, which meant the basal ganglia may have a factor, which meant, hey, maybe this isn't separate from dystonia. Maybe it really is dystonia. Dr. David Marsden, who started the Movement Disorder Society, really was the first person to say, yes, blepharospasm is a form of dystonia. In 1979, it was the first treatment with clonazepam. I know there's some things going on in the literature that says there's a risk factor for using benzodiazepines and dementia. I'm not sure we can say that is true. It would be like saying that birds come to the park when the ice cream man starts delivering. The ice cream man doesn't come to the park in the winter, and neither do the birds. And so just because you're on clonazepam, and you could be on clonazepam or another benzodiazepine a long time, doesn't mean you're going to get dementia. And I would tell you that in the literature for Parkinson's disease, there was a paper out of Australia many years ago that said Parkinson's, that 100% of this large Parkinson's population in Australia developed dementia within 12 years. Wow, I didn't want that out in the literature. Every patient I saw had a right to be worried about that. But some very smart people went back and looked at those data, and they found out that all of those patients were elderly. Now, elderly has to be above age 60 for me now, 56 really. But all of these patients were in their 80s when they developed dementia. And so when they went through and looked at that, it wasn't Parkinson's disease at all, it was age. So you have to be careful with this literature. And I think you need to not panic about benzodiazepines and the development of dementia. I'm using too much time with the sides. So the next big event, I think, in blepharospasm was a woman named Mattie Lou Koster, you may have heard of her, started the BEBRF. She began to then draw a constellation of people, researchers, but really anybody that she could find who was interested in blepharospasm was in her circle. Whether she liked you or not, you were going to be in her circle, and she was going to learn to like you. In 1982, that Wall Street Journal paper came about, and that really kind of changed everything. There was, maybe 3,000 people we thought had blepharospasm before that happened. And now we know there are hundreds of thousands. And it's important for you to realize that that was the work of just one woman who was mad that she was told she had hysterical symptoms. And it's amazing what the anger from a little woman from Texas can do. Then you heard today about Alan Scott's description and work with botulinum toxin. The first BEBRF grant went to Dr. Jankovic, and Dr. Jankovic used that money to prove that botulinum toxin worked for blepharospasm. So those 4 years that you were waiting for botulinum toxin to be approved by the FDA, he finished that work in about 1987. But without that double-blind placebo-controlled trial, we would not have botulinum toxin approved at that time. So here is your purpose. And so I want to kind of go through and talk about how you have lived up to that purpose. So if we look at clinical features of blepharospasm, we all kind of know all these. We've all worked with them. We know that it's eye blinking. We know it's involuntary. But we didn't know how to describe that and to really hang our hat on a lot of these things. And so early grants, maybe not so early with Dr. Baradelli, who looked more at neurophysiology, but certainly the grant to Dr. DeFazio began with trying to describe symptoms of blepharospasm and describe them so you could trace those symptoms through families. And also began to think about blink reflexes, which is what Dr. Baradelli was very interested in. And then we decided, well, there are a lot of risk factors. Do they make any sense? What if you have a family history of tremor? What if you have a family history of grinding your teeth? What if you have a family history of Parkinson's disease? What if, what if, what if, what if? And so I was working with Dr. Mahant, and Dr. Mahant was a fellow of mine at the time, and we decided we would start a survey. Mary Lou was very much in favor of this survey and helped us get it started. We didn't finish the work, but Beth Peckham did a fabulous job picking up where we left off, and we were able to demonstrate that many of these factors are the same factors we see in patients with blepharospasm. I think that's important because when you sit down with a doctor, if your doctor doesn't know these things, one, you need to think about, uh-oh, am I in the right place? But two, if you are, you need to educate your doctor to say, do you know about the BEBRF? The BEBRF is important to me and it should be important to you. The next is we all are worried about genetics. Well, genetics are complicated, and it's going to be unlikely that we find the blepharospasm gene, because what we're finding is dystonia can affect many people in many different ways. So if you look at that, there's a DYT1 gene you've heard people talk about. We used to think that everybody who had DYT1 had full-blown dystonia: legs, arms, trunk, eyes, face, neck. But that's not the case. We find that some people have blepharospasm. We find that some people have writer's cramp. And so I think it's important for you to realize that genetic testing is very important, and the BEBRF should be a part of it. But as I told Mary Lou last night, the BEBRF should be paying the I part, not the whole part. And I think you have to be critical with how you're going to use your funds, because they're valuable to you and precious to you to say, all right, I'm not going to give you $1 million to find a gene for all of dystonia. Now I'm going to give you 100,000 of it, because that's the part that really matters to blepharospasm. And I think that's the important thing that all these inroads that you've made with partnering with the other dystonia associations can allow to happen. To say, all right, we're in on this, but we can't fund the whole thing. We'll fund our part. So imaging was a big interest to us. If we could figure out functional imaging and what happens in blepharospasm, maybe we would understand it better. So Dr. Berman, who is an up-and-coming person in blepharospasm, I found some of his manuscripts, or I found, not his manuscripts, or I'd have shown you pictures, but I found an abstract of his that is very promising in terms of how he is going to use his grant money to really understand functional magnetic resonance imaging, or fMRI, and what happens in people who have blepharospasm. But you funded in 1990, with Dr. Perlmutter, an idea to see what sensory input does to the brain. And that came from an observation that people with hand dystonia or limb dystonia had an increased, you remember the Mick Jagger picture, that homunculus. So what the thought was, if you have an increased Mick Jagger face, you might be more likely to develop blepharospasm and might have a bigger sensory field. And that bigger sensory field would be more likely why you would get more information to your brain, which would cause you to have a bigger motor response. And so what he did was put a tuning fork around, and you could see that, I think the dotted, the stippled area is where the tuning fork was, and then you would look at the areas that were, the areas in the brain where the brightness is occurring. This was not a profound study. We were hoping to see that we would see a marked difference in these things and be able to validate what we saw in limb dystonia and other areas. But nonetheless, it was an interesting exercise to begin to think about the brain and the function of the brain in people with dystonia. You then funded John Mazziotto and then later his fellow who moved to New York to see what the difference is in brains and in the awake and a sleeping blepharospasm patient. And you can see it doesn't project very well. It looks a little better straight on, so maybe it looks all right over there. But if you look in the top right picture, that shows an area of the cerebellum and the pons that seem to be more affected in people with blepharospasm. So that led to the fundamental change in observation that dystonia is not a basal ganglia disorder only, because the pons in the cerebellum are not in the basal ganglia. So while we thought about the basal ganglia being the seat of the soul in terms of dystonia, now we're beginning to think, well, maybe that is not the truth, and we're seeing lower brainstem and cerebellar centers that seem to be affected. That led to a grant by that's coming later, Dr. Moorhead and the University of South Dakota, who began to think about descending control in eye blinking. And he advanced the field. I wish he'd come back, but he advanced the field when he was doing things. We spent a lot of time thinking about eyelid behavior and blepharospasm, and that is where you have a unique ability to study what occurs in blepharospasm, because the eye is a sensory organ that we can study much better than we can tactile discrimination in the hand for somebody with limb dystonia. So we've thought of squinting. Is blepharospasm just a lot of squinting? Does that mean you have a lot more photophobia? Do you have more pupillary reflexes to light, to sneeze, to yawning, to pain? Do you have a stronger blink reflex? And what about sympathetic drive? Do you just have more adrenaline in you, and that causes you to have more blinking? So you can see we spent a lot of time with that. So there are 5 grants that we began looking at that in general. And those are earlier grants that began to define things from a very regimented sort of thing. And then it came to grants that we funded later. The red star at the eyeball looks at that ciliary ganglion, looking at sympathetic control, and we funded that. And then we looked at the sensory organ itself from the eye to the optic nerve. So those grants have been funded, trying to determine the input and the sympathetic tone of the nervous system. But Dr. Hallett is really interested in the blink reflex. It's where he's put his career, and I thought it was a nice statement that Dr. Houghton made. He's back, I think. I saw him, about Dr. Hallett admitting, I know less and less about dystonia the more I know about dystonia. But you can see all of the grants that BEBRF has funded, all those little red dots. We have spent a lot of time looking at every nerve, every muscle, and every synapse, every connection in those pathways. Some people call failed data experience. We have a lot of experience with this, and I would say that we've not really able to come up with a way to deliver any information in that very complicated array of nerves. So if I were you, the BEBRF members, I would say, you know what, I'd like to spend my money in other areas. Is there any place new? Because I don't see any other spots that we can go in that pathway. And I think that is the fundamental purpose of what I'm trying to say to you today is you have to get educated and you have to figure out where am I going to put my money, because it is your money. You have to sell a lot of raffle tickets for it. So just in the next few minutes, I want to talk about treatment. We spent a lot of time on treatment. We've looked at brain, we've looked at medications. Dr. Rao talked about medications for the brain. We've looked at DBS a little bit, and I'll show you some work that BEBRF funded. Nerve resection, thankfully, we've not spent any effort in looking at how to resection nerves, except for what we heard today. Muscle, we've talked a lot about botulinum toxin. We've talked about how to reduce muscles, or funded that. One is a myectomy, just removing the muscle. But we had a strong effort led by a powerhouse in our society who's passed away from ALS, Dr. Wirtschafter, who was trying to look at how you could inject kind of a permanent toxin. So you would inject it and destroy muscle. So instead of having to have this surgery, which at that time was quite brutal, it wasn't near as good as the surgery we saw today. He wanted to look at injecting a chemotherapy that would destroy muscle cells to see if he could finesse that and kind of have a permanent benefit. We've looked at all kinds of visual and sensory modification, and I think that's why those C41 glasses work. If you have rider's cramp because you have a bigger sensory field in your hand, if you put on a C41 glass, maybe you've reduced that sensory field so you don't have as much sensory information coming into your brain to cause you to blink more. And finally, we've looked at a lot of sensory tricks. I don't see anybody with the glasses of sensory tricks, but there used to be glasses that you could weld on a little piece that would touch your eyelid and it'd be a sensory trick. I haven't seen those in a long time, so maybe toxin works. This is the work from Jill Ostrom and Phil Starr. Starr is the surgeon. Jill is actually, in my opinion, the star. She is an exceptional neurologist. This is out of UCSF. They had great preliminary data to suggest that this was a way to study this. BEBRF funded them, and here is their results. And so you can see if you look at eyes and the bars nearest to the left, you can see that at baseline on this Burke-von-Marsden dystonia rating scale, that the benefit in the eyes was better than what was seen in the neck, a little bit better than in mouth, and I don't know what maybe spasmodic dysphonia. She was able to demonstrate that if you, in the diagram on the left are the basal ganglia, and that's where she put the electrodes. And she was able to demonstrate that there was an effective, a point of maximum effectiveness to put those electrodes to get an improvement in blepharospasm and cervical dystonia. So we now have data to say, yes, this really works. So I'm much less reluctant to send people who are refractory to all other treatments for dystonia today to DBS than I would have in the past. Now let's say you have just blepharospasm. After seeing the presentation on myectomy today, I would be much more likely to send you to an operation for myectomy for just blepharospasm than I would for you if you had blepharospasm and cervical dystonia. That would be when you'd want to go to DBS. And so it's important to have somebody evaluate that and work through all of those things with you. So I've talked about the FL41 lens, and you can see the FL41 lens screens out a certain point of a certain wavelength of light. And so if you're thinking about how the rod cells or the retinal cells work, the less light input you have to those retinal cells, maybe the less likely you're going to have blinking. And many of you have noticed that because sunglasses really do help you. And these FL41 lenses seem to, at least in some people, screen out the light that seems to be most bothersome to them. And they, Dr. Katz and Dr. Degra have looked at all spectrum of light trying to identify these spectrals, and they keep coming back to say this is the critical point. But botulinum toxin is really the treatment, the first treatment of choice. This patient was seen by Dr. Jankovic many years ago. I've not seen somebody with this severe blepharospasm since I trained with Dr. Jankovic back in 1990. And so I think that that's also the work of the society, that people don't have to wait till they are functionally able to do nothing. And botulinum toxin can really make a difference. And you can see there are many, many grants that have come out of the BEBRF to look at how to make this toxin work longer, how to make it work better, can we come up with new toxins. I would tell you that it's important to me to kind of change a vocabulary. So I put this up originally because it was just a shameless self-promotion. But botulinum toxins are different. And if I'm going to say there's a difference in muscle between a levator and between a masseter, how come I can't tell you there's a difference in toxin? And the reason I think that's important is if you got injected with a toxin and you went to a new doctor because that other time didn't work and you said, well, I got injected with a toxin. They're all the same. They are not the same. So I would want you to use the terms. Won't make you use generics, but Botox is not Myoblock, is not Dysport, is not Xeomin. And so you need to be, to raise the expectations for yourself, for the society, and for your physicians. I realize it's easy to say Botox because I've been using the term Botox since 1990. But I don't use that term when I'm talking about Xeomin. I don't use that term when I'm talking about Dysport, and I don't use that term when I'm talking about Myoblock because I respect my craft. And you need to respect that also. You need to say, all right, all these toxins are different, and there's a risk to me if I get a shot with Myoblock and I get 2,500 units. And I go to a new person who's just read about toxin and said, oh, I got Botox, 2,500 units. Then you get to go to the ER, and then you get to worry about all of those poison things from botulism. And you get to get intubated, and you get to get all these things while you wait for that to wear off. So it's important for us to use our vocabulary. That's why we are humans. So that's my only soapbox, I think. But this is a handout that I think is very helpful. At least that's what I hear is very helpful. I even have doctors who have come up to me and say, yeah, this is really helpful to teach me how I can at least start injecting. And that handout's available on the website. So download it early and often. It's like voting. So one of the things that I thought was fun about this was to go back and look at the first 30 years of grant funding by the BEBRF. So as you look at that, I tried to look at the different categories: treatment, pathophysiology, animal models. I didn't show any of the animal models work. I talked about genetics, imaging. We had 3 workshops, and I think the workshops are very important, as well as the board meetings, to kind of define where we are, where we're going, and what else can we do. And if you look, you have 36 institutions that have been funded across the world So I think we should do a little better at that, but I don't think we should have one grant for every country. I think we should still be very critical and very careful to spend this money wisely. And it's good to get momentum in 10 centers rather than no momentum in 100. If you look at the total grants, 60 grants in 30 years, and we're spending about $35,000 per grant. So what does that $2 million do for us? So that $2 million took us from having about 15% of all blepharospasm papers that were published in the world in 1985 to 1994 to now in 30 years, we now are responsible for more than 21% of all blepharospasm publications. So I didn't have the skill set or ability to figure out how much money was spent on blepharospasm research in all those other 80% of manuscripts, but I bet it was a good deal more than $2 million. And so what I think that the society has been able to do, or the foundation has been able to do, is bring a collection of people with blepharospasm and researchers with or people with an interest in researching blepharospasm, maybe all that's the same, and begin to define what is worth looking at, or what is important to look at, and in many cases how we can get people to look at this more critically and improve their grants. And I observe in my work at Duke, as I see the society evolve with more FDA regulation, which I support. FDA is a safety organization, and so I think they're very important. But I think that particularly people my age are losing track of all the regulatory requirements that are needed for somebody to be a careful researcher with human beings. So before I challenge you, I want to remind you that BEBRF investigators published 21% of the papers in the field in the last 30 years, that your foundation has funded everything from DNA to sunglasses. You've been very good at translating these scientific findings into something that's readable on your website and into action and education. You have remarkably improved awareness of blepharospasm by partnering with the other dystonia groups and by attending neurology and ophthalmology meetings where you can talk to doctors. You provide a rich resource for support for people with blepharospasm, people who have loved ones with blepharospasm, and the physician community. You have been good at developing blepharospasm care providers and providing those names to people. And you work well with key stakeholders in industry. And again, we're very grateful to the support from industry. I know we might have a few extra eye drops, but Mertz has helped this meeting immensely, and not just with eye drops. They have helped us fund this so we could all have this experience, and I'm quite grateful to them for that. But here are my challenges. You need to talk to your doctors. If you have a doctor in an academic center, you need to say, are you interested in blepharospasm? Anybody interested in doing some work in blepharospasm? Would you like to do some research in blepharospasm? Would you apply for a grant for blepharospasm? How can I help you get this information? Because we are beginning to run out of steam in terms of new young people interested in doing research in blepharospasm, and we need to regrow that pipeline. We have not done that responsibly. I am partially responsible. Dr. Mahant was a very promising researcher, and I didn't keep on her to keep at it. And we need to keep on our people once we're developed. One of the things I'm doing at Duke that I really like is taking promising young faculty members and fellows and putting $10,000 of support around them. Say, all right, what's your idea? Who's your mentor? No, not a good mentor. Let me find you a better mentor, and let me pair you up where you can come up with a good idea, and I'm going to put resources around you so you can write a proposal, you can write an IRB, and you can get all of your institutional approvals done, and then give you some data management so you can do statistics correctly. Because we've forgotten how to do that, or I never knew how to do that. I had people who could do it, and my statistics responsibilities were much less than they are now. And so I think that that's something that you might want to think about in the future as, do we have a course of 5 promising people a year? And we put $10,000 around them, whatever is around them, but we make them and their mentor come to this course, and we beat up their proposals for a day, and then we send them on their way, and we make sure they get this done. I think that will dramatically improve the quality of our proposals, and it's my sense that the proposals have dropped a little bit in the past years. Now, this year was not. We had really good ones this year, but the years before that were not as good. Second is I think you need to become more prescriptive about what you're going to fund. I think you need to be more prescriptive about, here are the things we're interested in in blepharospasm, and we want you to make applications in these areas. So you can align the interest of the faculty you're trying to support with your priorities. And then I think you have to continue to partner with the dystonia associations, specifically for genetic investigation and for image analysis, because they have a bigger stake in it, and they will be more likely to garner resources from larger pools of money. And finally, I think you should partner for training grants. And I think it's very reasonable for you to say, you know what, we know you have a fellow who comes in, he sees dystonia. So in my world, I see Parkinson's disease, in a movement disorder clinic, Parkinson's disease is half the population. So that other 50%, about 30% of it is dystonia. And in my case, I like blepharospasm, so at least half of my dystonia practice is in blepharospasm. So if I want to have a fellow to come in to train with me, you should say, well, I know you see 15% of your patients have blepharospasm, so we'll try to fund 15% of a fellow. And I think that way you can deliver expectations a little bit better to say, all right, if I'm giving you 15% of that salary support, I really want that fellow spending 15% of his time or her time looking at blepharospasm and thinking about it. So I think you have, over the past 30 years, accumulated respect, and you now have a responsibility to be able to say, this is where we're going, this is where we're headed, please come with us. And that's all the preachiness I have. I really love this meeting. Thank you so much for letting me come talk again.